Fred's going to pop a question.
Thanks, Carl. Last night at our pre-meeting, we talked a little bit about FDA rules on cost recovery. Can you talk a little bit about how you managed to treat the first 30 or 40 patients? Well, first of all, can you give us a estimate of the cost for producing the cart for each patient? And then how did you manage to treat the first 50 patients between your grant that covered some of it and the other costs?
Yeah, so the initial 3 patients, I was unable to get support for the National Cancer Institute to do these trials. There have been so many failures and disappointments. I had an R1 to develop the actual CAR and we did the preclinical studies. Then it was all philanthropy and foundations such as the Leukemia Society that allowed me and our group to make the vector, which it was the first time that a patient had ever had an HIV-based lentiviral vector. That single lot initially cost $350,000. And we treated 3 patients with it. Now we can make 10 times that. I mean, there's been no effort in scaling up any of these things of cell and vector manufacturing, as you know. So the initial actual cell culturing charges we have on our website at our cell manufacturing and vaccine production facility at the University of Pennsylvania. They're costing. It's about $20,000 per patient in cost of goods. That's not manufacturing like recovery of depreciation and so on, but of the actual cell culture goods and so on. So that's quite cheap if you look at this therapy could replace a bone marrow transplantation, which, as you know, can be many times that. So when you look at the fact that we give antibodies at this point routinely to patients and they're non-self-replicating, we give recursive additions and that we pay $50,000 to $100,000 a year, I think engineered cells could be cheaper, much besides more convenient. So now, I mean, our initial, after we treated 3 patients, we were out of money. We had to wait a year to treat anyone. And so now, then Novartis came in, and now we have pharmaceutical sponsoring, to pay for the clinical trial costs now. And so that has now happened at several institutions where, Celgene has gone and partnered with Baylor to do cell-modified therapy. It's happened at your institution. So we've now gone to a point where there is a handoff, I think. There was no handoff before for these kinds of cell-based therapies. So there needs to be industry just as there is for other standard therapies.
Okay, last question, Roy.
Yeah, I enjoyed your talk. I would agree with you that the PD-1, PD-L1 therapies have really transformed lung cancer. And the interesting thing is 3 or 4 groups are in phase 3, each developing a different biomarker using different antibodies, different cutoffs, measuring different things, either the tumor cells, the lymphocytes. So I wonder what you think that says about the biology, and also what are the policy implications as this moves forward?
Yeah, so Roy, that's a great issue. I had one slide that said biospecimen analysis issues. So it's more complex, and there's no, how someone measures PD-1 or PD-L1 expression is different at different institutions, whether they're restricting it just to the tumor cells or to cells in the tumor microenvironment. And this is the kind of thing that the FDA's companion diagnostics is going to have to figure this out, because right now, even standardizing a flow cytometry assay between different institutions is difficult. So that will be a major issue.
Okay, thank you again, Carl. The other area that that was picked as a growing area that is on everyone's mind is informatics and IT. And we're very fortunate that Warren Kibbe has agreed to talk with us. I think it's only a week since he joined the NCI to take charge of their center for biomedical informatics and information technology. He spent many years at Northwestern, and he's worked on a number of consortia and group efforts, including CA-BIG and others, trying to develop tools for all of us to use that will help us to assemble data and make sense of the clinical data and the molecular data and the individual patient data. Warren?